This is the slideshow for Chapter 8, The Cellular Basis of Reproduction on Mitosis. I had pizza last night, and then I had heartburn, but I bit into the pizza and, of course, I instantly burned the roof of my mouth and my lip. And in doing so, I just, killed some of the skin cells of my lip and some of the cells in my mouth, and now they need repair. Part of that repair will involve replacing the damaged cells, and those replacement cells will be provided by copying some of the nearby undamaged cells. Luckily, this is a small wound, and it won't take long for my epidermis to heal it, but it still involves producing thousands of new cells through cellular reproduction, also called cell division, and here we're looking at mitosis. At my age, the principal function of cell division is to repair and replace damaged cells. But another function of cell division is growth, like the growth that's happening at the tip of this onion root. You are built from approximately 60 trillion cells. And every one of those cells can trace its origin back to a single fertilized egg that was formed about 9 months before you were born. So every cell in your body today was formed by cellular reproduction, starting from that first cell. Cell division, mitosis, ensures that the set of genetic instructions, your DNA, that's passed on to each of your cells, it's complete and it's identical. Different kinds of cells divide at different rates, depending on their role. Some human cells, like the cells in your skin over here on the right, and the cells in your digestive tract, they divide every day. Others, like nerve cells, are so specialized that they never divide. This may help explain why brain injuries and nerve damage never heals. Cells that do divide follow a sequence of steps. The cell cycle makes me think of this old punk rock song by Gang of Four. In it, they're just screaming birth, school, work, death. So the cell cycle always begins with a division from another cell, and then it ends with division into two new cells, birth and death. Yes, there are two phases in between, but they are not school and work. They are the interphase and the mitotic phase, or mitosis. Interphase has 3 subphases, but 2 of them are when the cell is just going about its usual business, and they're called gap 1 and gap 2. During this time, your brain cells are computing, your muscle cells are contracting, and the cells lining your small intestine are making digestive enzymes. A cell normally stays in interphase until it receives a signal from its environment. Once such a signal is received, the cell then moves on to continue the cell cycle, usually moving into mitosis. My skin cells that didn't burn last night, they knew of the death of their neighbors, and they were preparing to divide as I slept. Some of the cells remain in interphase forever, like neurons that make up your brain and the nerves of your body. To continue the cell cycle and begin the process of cellular reproduction, a cell must first copy or replicate the chromosomes, pieces of DNA, within its nucleus. This is accomplished during a subphase of interphase known as the S phase or synthesis phase. Synthesis for making more DNA. During the S phase, every chromosome in the nucleus is copied. When a chromosome is duplicated, the 2 copies are called sister chromatids, and they stay attached to one another at a location called the centromere, kind of like a waistline. Imagine yourself standing in a room. You're a chromosome, and the room is the nucleus. Imagine that I copy you, producing an identical twin, and you and your twin stay joined at the hip. Just work with me, attached to one of another like conjoined twins. So you and your twin would make up a pair of sister chromatids. with the centromere joining you at the hip. You look around and you see that within the room of the nucleus, you are one of 46 sets of identical conjoined twins. Think about that. 46 sets of conjoined twins, each joined at the hip. These are the 46 sets of sister chromatids found in the nucleus at the end of the S phase. I'm sorry, I have to take a moment here. I'm sorry. But there's this fascinating museum of human oddities in Philadelphia, Pennsylvania. They have many preserved specimens, skeletal and otherwise, of conjoined twins and other human oddities. It's called the Mutter Museum. I have been there many times, and I had the pleasure of meeting with the curator, Gretchen Warden, who was a bizarre and fascinating woman. It's worth a trip if you're in Eastern US. Let's focus our attention on the second stage of the entire cell cycle, mitosis. The purpose of mitosis is to distribute those copies to the individual daughter cells. Remember, each new cell will be a clone of the original. The cell doesn't normally spend much time in the cell cycle in mitosis, but many biology instructors would take a lot of time explaining it. Mitosis, it's a continuous process. It's divided into 4 stages called prophase, metaphase, anaphase, and telophase, or PMAT. I like this diagram of mitosis for an overview or a feel of the process. Note that there are 4 chromosomes in our cell. Humans would have 46 chromosomes. And it seems as though these chromosomes are from mom and dad, and they have different colors. Prophase is the stage when the duplicated chromosomes, they condense and they become visible. You can see them up there in interphase that they're not visible. The nuclear envelope disappears. From each end of the cell, a network of microtubules or little tiny tubes forms. This network of fibers is called a spindle. Each chromosome attaches its centromere to a fiber in the spindle. Then the duplicated chromosomes, they start to move toward the center of the cell. This proceeds to metaphase, where all of the chromosomes arrive and they line up at the center of the cell on an imaginary line called the metaphase plate. Anaphase begins when the sister chromatids are separated from each other. That is, each conjoined twin is ripped apart from its partner, and the 2 twins move in opposite directions away from one another. This movement is achieved by motor proteins on each centromere that drag the chromatids along the tracks laid down by the spindle. Each chromosome is now a single separated chromosome, but duplicated from its partner. And we can distribute these copies to the new daughter cells. During the final stage of mitosis, telophase, the single chromosomes arrive at the opposite ends of the cell. On the stage of the 2 sets of 46 individual chromosomes, they arrive at opposite ends of the cell, and then they begin to disappear as they're enveloped by the nuclear envelope. The chromosomes unwind, they become invisible once again, and it seems as though we enter into another interphase for both of these 2 new cells. So we've achieved the goal of mitosis, 2 completely new cells, each with an identical set of chromosomes. Once mitosis is over, we still have to divvy up the rest of the cell contents, like the cytoplasm and all the little organelles. This process is known as cytokinesis. Actually starts as telophase is kind of like ending. The result is 2 new daughter cells, each with its own nucleus and each with its own share of cytoplasm. Cytokinesis in plant cells involves this cell plate down here on the left that separates 2 daughter cells. Animal cells form what's called a cleavage furrow that divides the cells. Here are the most important take-home lessons to remember from this tutor session. Multicellular organisms use cell division for growth, repair, and replacement. Most of the time, cells are in interphase, right? But then if they receive a signal, they duplicate their chromosomes and complete the S phase. Then they move into mitosis, and they separate their chromosomes and the cell, and then the result is two genetically identical daughter cells. Of all of the mitosis examples I have ever seen, I think this is the best one. Take a look at some of the detail on this diagram. It's great.